Good morning, everyone. This is Justin Borvitz. I'm going to be talking about the biology side of the Bioinformatics and Functional Genomics course, Biol3157, here at ANU. So this is an overview. We're going to have a practical on Friday about population genomics, and we'll go through samples from a population and sequencing-based genotyping to get genetic information across those samples. And the genetic information across samples can give you a sample by sample relatedness matrix, also known as the kinship matrix, as well as individual variants on all those samples. So those are information that can tell you about the relatedness. So the next section will be how is that genetic relatedness distributed on the landscape. Where are your progeny? Where are your siblings and kin out there in the world? And so the landscape is kind of a word that includes both the geographic coordinates, kind of a flat landscape, or even with altitude. And then there's, of course, the environment, whether you're close to the equator and it's tropical or temperate regions, and lots of other information that can be obtained from the GPS coordinate about the environment, which is correlated with geography, but not necessarily causal. So when we know about genetic variation in the landscape, we can try to dissect the neutral processes, isolation by environment, as well, isolation by distance, as well as isolation by environment. And then the last workshop is going to say, okay, we've got these samples, we know their genetic relatedness, but what do they look like? What are their disease states? And that's a phenotype. If they're plants, which we'll focus on mostly, how fast do they grow, and how do they grow depending on the environment? So the phenotype is, of course, a product of nature, which is your genes that you're born with, and nurture, the environment that you're growing up in. And we'd like to measure those phenotypes often at many stages throughout the plant's life cycle. And if we could put those same populations, or if we know how the populations are related, we can grow them in different environments. And then scan through to see how much of the phenotype is explained by the background relatedness and how much is explained by individual genes. And that will complete this section. So you'll see genotype to phenotype and environment. And this omics is kind of what you're learning a lot of these days, big data. It's many, many genes, many, many phenotypes, and many, many environments. And we're thinking about integrating this three-way puzzle piece, even all the way to the globe, and think about how populations move around the world, which is exactly what agriculture is. Okay, so let me jump right in. I'll give you the story about Brachypodium. It's a mixture of multiple species. Part of the interesting thing about doing genetic analysis is just to get not only the populations and who's related to who, but to dissect cryptic species, which sometimes things look the same and they're really not the same species. Sometimes there's interspecific crosses and polyploids that result. And a compilospecies is a mixture of different species in a species complex. This little grass is a model for wheat, and so that's why we got into it. And one of the first projects we started when I came to Australia was to go out, try to find these weeds, and they can reproduce really quickly and spread around, and so we thought it'd be a good model for adaptation. Of course, to be a good model for adaptation, you need genetic diversity, and you need recombination events to shuffle that genetic diversity so we can figure out not just who did the best, but which individual genes are allowing you to do the best. And so we first needed to go around the world and collect samples, and that's what the data is that you'll be analyzing in this class. Jared Strike was a very talented PhD student. He's back trying to finish his thesis now, and Pip Wilson, the postdoc, Kevin Murray, who you know. Everyone contributed to this project. It's a big international collaboration. So the idea is to get the genotypes, get the set of samples that would represent a diverse collection. And if you have a whole bunch of collections that really don't contain genetic diversity, you're really wasting your time managing huge sets of what are known as accessions without the genetic diversity. So these days, everyone in stock centers is racing to inventory their true genetic diversity in their collections. And oftentimes there's multiple accessions that have been passed around and resubmitted, and they have the same genetics, maybe a few rare mutations, but essentially the same background variation. And people then try to grow those up and look at the phenotype. When you understand the phenotype-genotype map, which we'll talk about on the very last day, then you can reverse this problem and predict the phenotype from your stocks and your seeds. And you might be able to even do that across different environments so that people can order seeds from a collection and have a good idea about what they'll grow up to look like. This is tapping into the ancestral genetic diversity, usually within species that it may not have ever even grown out these particular genotypes before, but just based on the relatedness of samples that have been grown out, you can predict the phenotype and what they look like. So that's a revolution in breeding. It's the genomic selection approach, and it'll really be a powerful way to accelerate breeding in perennials and things that you don't have to select every single year, that you might need a decade to look at what their growth is. All right, so of course the growth then is dependent on where you grow them and how you grow them. And sometimes we can look at the distribution of genetic diversity across the landscape and learn about what was in the past an adaptive genotype. So you'll see both the neutral process of how populations are distributed across the landscape, and you'll be able to also scan for adaptive genes. So this gene pool then, where they come from, is an indirect phenotype on their fitness in those environments. Or the fact that I found you here must mean you can survive here. Now there's a huge kind of false negative in the genotype environment or genotype landscape axis, because not all genes and not all populations have been tested in all environments, right? So we only know about the success stories. It's not that all genes in all populations were tried in every part of the world, although maybe fungal spores and certain things can span the globe and the environment filters. So within the range of neutral gene flow, you can have selection on adaptive alleles, and that's called landscape genomics. And once you understand that and the environment shifts, or you want to move things around the world to where they've never gone before, you can predict what environments would be suitable for these genotypes, just in the same way you can predict the phenotype from plants that haven't yet been grown. So this is an important paradigm as well. Now, what do we want to plant where? That's an old question that's predating this genomics revolution. We want to know, what agriculture should provide. And early on in Australia, the idea was to cut down the trees and put out sheep for wool, send it back to the UK, and a lot of that legacy agriculture still exists today. It's not necessarily the right thing to do for the soil or for the future. But genetics is a tool that can allow you to predict where things will do well and what traits will do well, even if that hasn't historically been used. So there's a big maybe social problem to figure out what should be grown where, but once you've solved that, then digital breeding can really accelerate the process. So Brachypodium kind of comes from Mediterranean regions. It's very similar to the wheat growing regions of the world. It's distantly related to some of the major crops, wheat, barley, and rye, as well as several grasses that are pasture grasses. And it's really hardy. Basically, it can reproduce quickly after a rain and then sit as a dormant seed, and the little awns can get stuck to animals and get, so it's a fantastic migrator in practice. It gets stuck on animals and they get traveled great distances. There's also kind of an overwintering life history strategy where you reproduce later. You germinate in the fall and overwinter as a green grass, and then when things start to get dry, they put up quite a few more flowers. So that's another little Arabidopsis plant nearby for comparison, which is more temperate and lives in cooler climates. So Brachypodium has been introduced to Australia and reported in the Atlas of Living Australia at many locations. We never made it out to Perth to sample, but we did catch a lot of new accessions around Adelaide and between Canberra and Adelaide. So we added some local accessions to the collection, as well as many from around the world. So the native range is the Mediterranean, sort of east in Turkey and west in Spain, and depending how you sample, we're starting to learn after years of staring at the data that just collecting on the roadside may not be the best way to get genetic diversity. That it's important not only to get geographic diversity, but to really get off of the track and into some less disturbed areas where there could be some older populations of diverse Brachypodium. And we found a few in Spain that are worth recollecting, but we sort of got the weedy types, the cosmopolitan types, because they're the ones that have done well in agriculture and made it around the world. So the list of accessions is close to 3,000 from nearly 1,000 locations, but how many genotypes did we have? And that is a question that we used second generation sequencing for. We used genotyping by sequencing, which is a reduced representation approach. And if we were to do this again, we would use these pocket sequencers, which I can tell you more about another time. So the idea was to take all the short reads that come out of the sequencing approach and line it up against the reference genome. So we'll talk about this in a little more detail, but the idea is, and so you can do this yourself, the idea is that you count the number of reads that match to either genome, and if you've got a compilospecies, if there's multiple species in there that are related to the two reference genomes, then when you've got pure species Brachypodium distachyon, all the reads will essentially match one of the reference genomes, the distachyon genome, and if it's a Stasii sample in red, most of the reads will match the Stasii genome, and if it's a hybridome, which is this polyploid between both genomes, then this sample, which is pretty similar morphologically, it's slightly bigger plant when you get to know them, but out in the field, you really can't tell. And so the hybridome's got both genomes in there and reads aligned to both of them. It's pretty clear with the genomic signature which species it is, but it's not so clear phenotypically. So this is the first step to unraveling the genetics, is just to get the species right. All right, so here's the distribution around the world. What can we learn from just the species? So it looks like, except for one maybe collection in Australia, all of the global invasive types are Brachypodium hybridum. There's a little bit of Stasii in both the east and west native range, but Brachypodium distachyon is essentially limited to the native range. So both of the little diploids are still in Europe, and the polyploid has gone global. And this is not unusual. In hindsight, of course, that's maybe what you'd expect, but there's precedent from other species about how fusing two genomes can give you some hybrid vigor or overdominance that allows you to be, really successful around the world. Now, the problem is that you can limit your adaptive potential if you're successful invasive and you have limited genetic diversity and you're going about the globe. How do you, you might be doing well today, but how will you do well when the environment changes? What sort of adaptive potential do you have? So we're going to now look at the genetic diversity within each of these species. So this is the count. So like 1,000 accessions were hybridum, about 500 were Distachyium, and 50 Stasii. What's the genetic diversity within those samples? Well, this is a map of the Israel and South Spain actually are mixed for Stasii, and maybe Israel is in a place where you could find new polyploids happening. It certainly could be that if there are multiple genotypes of Brachypodium hybridum, that they're the result of multiple hybridization events rather than, or at least two hybridization events independent that then came together and released some genetic diversity. Otherwise, they'd be all one big clone. So let's take a look at that. And this is hybridum in the introduced range. Let me just talk about this theory slide for a second. We wrote this paper called "The Missing Heritability is in the Field," and it was to try to address the serious confounding that these new genome-wide association studies were having with population structure. And so after the Human Genome Project was finished, the medical community went to genome-wide association studies to try to get at the complex genetic basis of common diseases, and they basically failed after many hundreds of millions of dollars in studies. And why did it work so well in plants? So the idea is that plants just have huge families. They have hundreds of thousands of offspring, and we could look at how the genetic deck has been well shuffled in plants and get at the cause of genetic variation. And we used another trick is that sample selection, sample study design, being able to take the most informative subset of accessions from a large gen bank. So if you have a stock center with tens of thousands of accessions and you can look genetically at them, this across the top is there's going to be structure inherently due to biases in sampling and migration and just the non-random history. But if we want to do common garden, detailed phenotypic studies where we're investigating the health and development of every individual sample, we'd like to do that in the most powerful way. And so if you can focus on certain populations, which there's certain human populations where people focus on, certain parts of the landscape where you can collect wild populations and do a regional mapping approach where you say, look, there's a lot of variation out there, it's structured, but I'm going to focus on some recent admixture or local populations. That's one approach that has been successful. The other is to do these synthetic populations, which I call population restructuring, and that is to just take all the long branches, all the ancestral genetic diversity, and then cross it together. These are synthetic populations. Breeders do these all the time. It's a series of wide crosses. And then you can look at the release of all this cryptic genetic variation within species groups between long lost relatives, if you will. So by shuffling the deck and balancing the cards and then reshuffling it, you can get a powerful study design that allows you to dissect the causative genetic variation from the correlated background variation. So that's population restructuring, and we wanted to do that to the Brachypodium set. So these are three observed trees. Let's look at the bottom one, hybridum. There's 1,000 accessions, but only about 80 genotypes. And if you look at those long branches, there's very few long branches and a whole bunch of short, sort of noisy branches. And we pick a threshold for clumping technical replicates. Two seeds from the same packet, and these are plants that are self-pollinating, so we expect them to be largely inbred. Two seeds from the same packet are essentially biological replicates, or two leaves from the same plant would be technical replicates, and the genotypes of those replicates should be identical. but there, no sequence reaction is perfect, and in these high throughput approaches, there's sometimes some noise. If you get a sample that doesn't work very well, you might not be able to say exactly what it is, but you could say it's closely related to these other things that I've also looked at. So that's basically explaining away much of the noise down at the very tips of this genetic dendrogram, or sort of a representation of an intra-species. Phylogeny is not the right term here, because there are ancestrally recombining approaches, but recombining chromosomes. But once they become self-pollinating and move around the world on their own in these sort of invasive patches, there's not a lot of mixing happening. And those old branches are the legacy of original outcrossing. And so there's a few deep branches, and those would be considered different genotypes, and then their migratory histories can be tracked individually. So it's a very powerful approach to managing invasive species, if you can say, oh, here's another one, but we know where it came from because we've seen these genotypes before. So it's very high clonality here. It's the ratio of accessions to genotypes is more than 10 to 1. Whereas if we look at Stasii, there was 50 samples and 10 different genotypes. So that's a little more diverse, but it's a very small sample, and maybe that species is going to get wiped out. Well, it's a good thing we've got some in the stock center. The Stackian, that's the reference genome that paved the way. We didn't know that there were other species when this whole game started, but there's 500 accessions and 130 genotypes. So we're very limited on genetic diversity in the diploids especially, but that's what you get when you study these invasive species. So very low outcrossing, lots of migration, and lots of resampling of similar genotypes. So that was the end of a big study. Now we can look at, and you guys will generate the first pass analysis here. We went to the core set and did whole genome sequencing, which is kind of covering the 200 to 300 megabase whole genome data at about 10x coverage. So it's a little more money in sequencing, but nothing compared to the amount of time we've spent on this. Now, the red blocks are pairwise correlations between genotypes at almost 2.5 million common variants. So it's 100 by 100 kinship matrix here. There's a few samples in white that are quite different from the other things. WLE2, maybe a different subspecies within Dipsacian. And then there's a whole bunch of these red clones, the BDTR, 1 and 2, H, B, C. So some of these are the biological replicates that we expected to be very similar. All right, now if you make a dendrogram of that, it's very easy to tell. Some of the technical and biological replicates are clustering right next to each other, and then there's two main groups of the Stackian. That's with a couple outliers here. So Cas2, WLE2, and BD1-1 are three samples that are in this other subspecies, and then the first branch then splits into kind of a generally east and west in the Mediterranean, but a lot of technical replicates within. So we tried to minimize that, to not spend extra on resequencing deeply things that are actually the same, but some of these replicates carried through, and that gives us confidence in the distance that we're looking at. Now, when you use whole genome data, the signal is like night and day, right? It's millions of variants, so we can see the very low diversity within a genotype and the medium to high diversity between genotypes and subspecies if you look on the y-axis. All right, so that's how we kind of collapsed these redundant ones and excluded the outliers. That's just QC approach, and now we're down to about 60 samples, which is actually really low, but they're at least diverse, highly diverse samples. So the effective population size might be 3 times that or something. So these core collections are very powerful in that there's better than random amount of diversity. The effective population size assumes that things are randomly mating, which is a ridiculous assumption because they're mostly inbreeding. However, it's still, with this selection process, your actual population size is better than would be expected from random mating even. So there's the dendrogram to go with that. Now, what happens if we turn this problem around a little bit? And with whole genome data, we can start to look at the block size. And so now we're not going to compare sample by sample to figure out overall relatedness. We're going to compare site by site. So let's take all these samples, which are actually different from each other now. They have some shared history, but they're all independent recombinations. So we can look at the correlation between SNPs going down the chromosome. And you might remember from second year genetics that variants on different chromosome arms should essentially be unlinked, and variants in different chromosomes should be unlinked, and that once you go through one round of meiosis, they're inherited essentially independently, right? Because chromosomes get independently assorted into the progeny, and long arms and short arms of different chromosomes each through at least one recombination go their separate way. Well, if you go back hundreds of generations to the history of these Brachypodium samples, there'll be more and more ancestral recombination and until you get to sort of the finite state, and that's at this red crosshairs. And so different parts of the chromosome have different amounts of recombination. Sometimes it's 5 to 10 gene size, kind of 25 to 50 kb. In other parts near the middle of the chromosome could be really long blocks, almost quarter of a megabase or even larger to a megabase. And so these repetitive regions don't get a lot of shuffling. But this is our best approach now to get at the functional genetic basis. So selection in the environment is acting on specific genes rather than background populations, as well as the traits that they control. So once you have a recombinant set, you can start to look at the gene's effect independent of the background, independent of the lineage that they come from. Right? So that's a twist. There's really two ways of looking at this. One is how the samples are related to each other. That's the neutral processes. The other is how the variants, the SNPs, are related to each other. And that's where you start to see selection acting on some genes as opposed to others. All right, let's talk about how to grow these plants. Now that we have our core set, this is wheat growing outside, and the days are warm and the nights are cold, and depending if you're growing in Queensland or New South Wales, it'll be different temperatures at different times of year. So we set our chambers to mimic the field without the weather, and that's how we can try to get at the growing conditions. And we call these climate chambers, and through 2018 we'll be building 4 climate containers, each with 2 rooms that can simulate different parts of the world, different growing seasons, spring, fall, north, south, east, west, anything you want to set in there. And then we can look at the predictive, what you might expect from an average year over a decade kind of type of experiment, rather than any one location in one year, which is filled with anomalies of a particular trial run. So these are going to give us a lot of flexibility, and if you're interested, come talk to us more about helping getting this set up. But we should be able to look at thousands of plants seedling stage and hundreds of plants at the adult stage. Brachypodium is so small that you could do thousands all the way to adulthood, which is one of the real advantages. Special lighting conditions that allow us to simulate the field is all very critical. My PhD is on light response, and so leaving these spectral signals out just confuses the plants on what time of day it is and what time of year it is. And so putting them back in gives you a really good match from the lab to the field. And we can image the growth in multiple colors Here's some movies showing you how the chambers work, including the diurnal shifts. Let's see if they play for you. Might take a break and see how the recording is going. So these videos should play. That would really help. Okay, let's stop here for now.